So now we're going to talk about peptides, which are molecules that have amino acids joined together. A tripeptide has 3 amino acids, and the amino acids are linked by what is called a peptide bond. And that, of course, is right here. So we have that carboxyl terminus of one amino acid reacting with the amino terminus of the next amino acid. So this is that amide bond. So we can classify peptides by the number of amino acids. So we've used the prefix tri to indicate we have 3 amino acids in this peptide. Polypeptides have many amino acids, but we could have a dipeptide, meaning 2. We could have a tetrapeptide, in which we have 4 amino acids. Once we get above 10, we generally call those oligopeptides, and greater than 20, maybe 20 to 40 polypeptides. Proteins, however, will have more than 40 amino acids in them, and that is necessary for them to form their structures properly. So here we have 2 amino acids, alanine and serine, and we see their functional groups, the C-terminus, or the left-hand side, which has that carboxyl group, and the amino term or terminus of that serine. And these 2 are going to react. We're going to release a molecule of water, which is from the oxygen on this carbon and the 2 hydrogens on this nitrogen, and form that new amide bond, and of course release a molecule of water. So order matters. So the order of these peptides in a protein or a polypeptide structure is going to contribute to the folding of that protein or polypeptide and to the activity. So order matters. In this case, we're combining serine first with alanine. So when we talk about the sequence of a protein, we're talking about the order of the amino acids within that structure. Okay, and it does make a difference. So I've already said these words before, but the N-terminal or N-terminus is going to be the amino acid that is on the left, or the furthest to the left, and it has a free amino group. The C-terminus of a peptide is going to be the last amino acid on the right, and its free region is the carboxyl terminus. And we can name these simple peptides that only have a few amino acids by just linking their names together. Well, the first amino acid in the name ends in YL, and then we get the full name of the second amino acid. So this would be alanyl serine, or AL-SER, and this would be seryl alanine, SER-AL. So these are the 3-letter abbreviations for those respective amino acids. So what is primary structure? This is the sequence of amino acids in a protein. And as I said, order matters. It's going to dictate how that protein folds and contribute to its activity. Okay. This is a ball and stick model of a polypeptide that has 4 amino acids, so this would be a tetrapeptide. And you see that all the bond angles are 120 degrees, giving it this zigzag arrangement, which is actually as a result of that backbone of the nitrogen, carbon, carbonyl groups as they link up. Secondary structure of a protein is going to be the 3D arrangement of those amino acids in space. So there are intermolecular forces that contribute to these arrangements, and the strongest and most important one, of course, is hydrogen bonding between the NH group of one amide with the carbonyl group of another. So this is going to be one of two stable arrangements here. Although there are some others that we won't talk about. But the two structures that arise due to hydrogen bonding are the alpha helix and the beta pleated sheet. Most proteins have regions of both alpha helices and beta pleated sheets, and there may be other regions that are random, so there's no organized structure to those regions and no hydrogen bonding between amino acids in that region. So this is what alpha helix looks like, and you've probably seen this if you've studied DNA. These are the hydrogen bonds. So we have the carbonyl group on this amino acid, hydrogen bonding with that hydrogen attached to that nitrogen up here. We call this the rise, and the rise here for one turn in this helix is 3.6 residues. Residues residue is another term for an amino acid. The R groups, as you notice, stick out from the sides of these, this structure, okay? So they are all on the outer edge rims, and the backbone is in this curved region. This is a depiction of a beta sheet, and you'll see that this up and down structure or shape of this structure is the pleat. There are two forms. There are parallel and anti-parallel beta pleated sheets, but you also can denote or pick up the hydrogen bonding here between the sheets in this structure. So here again is a nitrogen group from the first amino acid in this sheet that is hydrogen bonding with a carbonyl group over here. And then you have the reverse. We've got the carbonyl group interacting with the hydrogen on that nitrogen, forming that hydrogen bond, which stabilizes this structural arrangement. So secondary structure is based on hydrogen bonding and can be either alpha helix or a beta pleated sheet. So these are just some ways that we denote what that secondary structure is. So these are shorthand symbols that you might see in a protein ribbon diagram. This represents the beta sheet, and of course we have this helical ribbon that describes the alpha helix, and this is a random region in a protein. So this is a ball and stick model of a protein. So we can see the actual carbons and nitrogens, side chains, and while the ball and stick model gives us more information about the atoms and types of atoms that are present, it is difficult to see the actual secondary structure that's present. So this is a ribbon diagram, and that allows us to see where the alpha helices and the beta sheets are in this protein, as well as the random regions in 3D space. So this is the secondary structure of silk, and it's organized, obviously. So this is a single strand of silk, and we see that we have alpha helices, we have some random regions, and this ordered beta pleated sheet that's central here. And keep in mind that silk is a very strong molecule. Tertiary structure then is going to be the 3D shape of that molecule that comes from the way that molecule folds and orders itself in space. And it is adopted by the entire peptide chain. It is driven by the maximization of hydrogen bonding with water, and the nonpolar side chains are stabilized by London dispersion forces on the interior of the structure. So remember, if we have our cellular components are quite a large percentage water, so if we have, in general, the exterior, of course it depends upon the tissue. If you're in fatty tissue, it's different. But in general, the outer regions of that protein are going to have the polar or more polar amino acids that will interact with water, and the nonpolar amino acids will be on the interior of the structure. Polar functional groups can hydrogen bond to each other. So remember that some of those side chains have polar functional groups. So there will be hydrogen bonding between side chains, as well as other types of interactions, such as electrostatic interactions. So some amino acids, as we've seen, can have positive or negatively charged side chains, and those will interact with the partial negatives and positives of other amino acids or the surrounding water. Disulfide bonds are another type of interaction that contributes to the stabilization of tertiary structures, and these are actually covalent bonds between two sulfur atoms in different amino acids. So here we have a depiction of disulfide bonds, and here we have side by side two amino acids with that sulfhydryl group on its side chain. So that's similar to a hydroxyl group, only it's a sulfur instead. And when this is exposed to an oxidizing agent, we can get the development of a covalent bond here between those two sulfurs within the same polypeptide. Or we can have the interaction of the sulfide or thiol groups, another word for that is thiol, between two different chains. Okay, so these are different polypeptide chains. So this is an intramolecular sulfide bond, and this is an intermolecular sulfide bond. But both have the formation of that sulfur to sulfur covalent bond. So this is an example. I think this is only one polypeptide, but it shows some of these interactions that we've just been talking about. Here's a hydrogen bond between a carbonyl carbon in the backbone with hydrogen that's bonded to amino nitrogen in the backbone. Here's an interaction between an amino group in a side chain with a carbonyl group in the backbone. Hydrogen bonding, of course. This will be an electrostatic attraction. This oxygen is negative, and this amino group is positively charged at physiological pH. So there will be a plus-minus attraction here. These are both nonpolar side chains, so we have London dispersion forces interacting between those over here as well. This is an intramolecular disulfide bond. And here we have the interaction between hydrogen bonds forming between these hydrogens that are bonded to oxygens. And here we have an alpha helix. So this is what some of those interactions might look like. Quaternary structure requires 2 or more polypeptide chains. So this is the interaction of, or the shape that is adopted when these different peptide chains fold together and make one complex. Insulin, for example, consists of 2 separate polypeptide chains, and they are linked by intermolecular disulfide bonds. So you see in the first chain, or the A chain here, residue number 1 is a glycine, and it has a total of 21 residues. And in the B chain, we've got a phenylalanine as residue 1, and a threonine as the last residue in this chain. And you see there will be interactions between, or intramolecular interactions between these 2 parts of the chain. But we have disulfide bonds between these 2 separate chains. So we started with primary structure, which is the actual amino acid sequence in our polypeptide. We have seen secondary structures that arise from hydrogen bonding. That can be the alpha helix or the beta pleated sheet. Tertiary structure, the 3D shape of that protein, so how it folds and arranges itself in space. And then a quaternary structure is a complex of polypeptide chains. And in this, it looks like we have 4 different polypeptides here that are interacting to make this quaternary structure. Hemoglobin is an example of a protein that has quaternary structure. Myoglobin, a relative of hemoglobin, is just one polypeptide chain. Oh, yep. So here we go. Hemoglobin and myoglobin are globular and conjugated proteins, meaning they contain both a protein and a non-protein component. Also examples of tertiary and quaternary structure. Their non-protein unit is a heme group, and this is a complex that surrounds an iron 2+ ion. Okay, so hemoglobin. Iron 2 plus is responsible for binding O2 gas in the bloodstream. So hemoglobin picks up oxygen in the lungs and then takes it to the cells in our tissues where it's exchanged, transferred to the tissues. Well, I've already said this, hemoglobin transports O2 to wherever it's needed. So hemoglobin can actually bind, there are 4 subunits, and there are 4 heme groups, 1 in each subunit, so it can transport 4 molecular oxygens at one time. Or, if needed, myoglobin stores O2 in the tissues. So it's a little bit, enables us to reserve a little bit of oxygen. This is what that heme group looks like. Here's our iron 2 plus, and it is what we call coordinated by these nitrogens in that structure. So this is the heme unit. Myoglobin has 153 amino acids, and as I've said before, it's one polypeptide chain. And here is that heme group, and where it will, so that's the iron, and that's where the oxygen gets coordinated. Hemoglobin, as I've said, has 4 polypeptide chains, 2 alphas and 2 betas, and thus those 4 heme groups, 1 in each of those subunits. And of course, whereas myoglobin has only tertiary structure, hemoglobin has quaternary structure. So the heme group in either hemoglobin or myoglobin, but let's focus on hemoglobin, has a particular affinity for oxygen. And it binds oxygen with what we call a cooperative effect. So once one oxygen binds to a heme group in one of the subunits, the other binding regions or heme regions have a higher affinity for oxygen and bind oxygen molecules more readily. However, when it comes to carbon dioxide, and this is what makes it so deadly, it is bound 200 times more tightly to the iron group than oxygen is. And once hemoglobin has complexed with carbon monoxide, it can't pick up oxygen. And of course, cells will die from lack of O2. So that's why the treatment, whoops, I'm sorry, I didn't realize. I thought there was something else there, so sorry about that. The treatment that thus then is, if caught early enough, is to saturate the tissue with oxygen, your body tissues with oxygen, to force that carbon monoxide off of those heme units. Sickle cell anemia, you've probably heard of. It's a defect in a red blood cell, and there is just a single amino acid difference in 2 of the subunits of hemoglobin, but that can have a deleterious effect, obviously. And I said the red blood cells are misshapen. They become elongated and crescent or sickle-shaped. That's what the name how it's named. And these red blood cells can rupture capillaries, which leads to pain, inflammation, we can have organ damage and even death as a result of sickle cell anemia.